All right, first, thanks, Adrian. This is a really cool opportunity and I think a great success for the field, and I'm really excited to participate, and thanks everyone for tuning in. I'm going to talk about a particular type of navigation in C. elegans and a neural circuit we think is important for it, but I want to contextualize it into a bigger question that really fascinates me in the lab about perception and behavior in general. And that's related to how we integrate our own movement and behavior into how we perceive the world. And it's going to be important for the talk. And this is work I started as a postdoc with Yun Zhang, and in my lab it's being done by Marie-Helene Hulette and Lily Guinet primarily right now. And so this is sort of a combination of all these stories. So when we look at a beautiful painting or a visual scene like this, our eyes kind of naturally flit around it through these rapid movements called saccades. And so actually the features of the painting, the colors and the textures and all the photons are constantly shifting where they're being projected on our retina. So they're activating different subsets of retinal cells and retinal ganglion cells and different populations of neurons in our primary and secondary visual areas. But despite all that moment-to-moment change in which neurons are representing the scene doesn't appear to move to us. It appears sort of stable and fixed in space as our eyes move around. We can get at why that is to some extent by doing a kind of a home experiment. If you signed your waiver, you can cover your eye and sort of push your eyeball a little bit from the outside. So be careful, please. But if you do that, what happens is most people see something that kind of looks like this. The image in the visual field does kind of move. You perceive it moving when you push your eyeball with your finger. And so the difference is whether your ocular muscles are sort of controlling your eye movements or whether something external or as external as your finger are controlling them. And this is really gets at something that our sensory systems do almost invisibly. So our representations of the world stay stable even as we're moving and interacting with them. And this is completely effortless to us, but it requires that our brains are constantly determining whether sensory information is occurring because of our own behavior or if it's occurring because something is happening in the world that we need to care about. So this is a complex problem, but to me it's interesting because no matter how simple an organism you are, this is a useful thing to be able to do, to have this distinction between self-generated stimuli and externally generated stimuli is very important. So depending on the scientific or philosophical tradition you come from, people have different words for this phenomenon. In ethology, it's often called corollary discharge, which is the term I'm going to use. But the way it's thought to work and has been thought to work for a long time without a lot of good examples is that when an animal has an intention to move and produces a motor command, this produces an action. This action produces reafference, which is the stimuli caused by the animal's own behavior. And a combination of this reafference with exafference, which is everything else happening in the world, produces the actual feedback the animal perceives due to its behavior. So at the same time, at this motor commands issue, there's a copy of that command, which we're going to call a corollary discharge here. And this corollary discharge is used to produce a sort of predicted feedback, so prediction of what the sensory consequences of the action will be. And then what brains do is they compare this predicted feedback to the actual feedback to try to distinguish between self-generated stimuli and exafferent stimuli, and to do all sorts of other interesting sensory motor computations. So the simplest thing you can do is the actual feedback minus the predicted feedback should tell you what the exafference is versus the reafference. But there are very sophisticated systems that have been studied in ethology, like echolocation, that use this kind of system where maybe a dolphin squeaks, that produces the action command to produce the squeak, but also a prediction of what that squeak will sound like, and a comparison between the predicted feedback and then the actual feedback, which includes the squeak, but also includes all the echoes coming off objects in the environment, is used in a comparison to produce a spatial image, basically, of the environment. So these kinds of active sensing mechanisms often employ, or are thought to employ in most cases, systems like this. And I think it gets more basic because a lot of sensing that we don't normally think of as active sensing really is in this sense. For example, olfaction involves breathing, so your diaphragm moves up and down to draw air across your olfactory epithelium if you're a human or a mouse. And there's actually a signal from the motor system that controls the diaphragm to the olfactory bulb so that the bulb can modulate its responsiveness depending on whether it's an inhalation or exhalation part of the cycle. Similarly, I was sticking with rodents for now. Whisking, which is my favorite rodent behavior, where animals move their whiskers back and forth to probe their environment. There's a signal from the motor system controlling protraction and retraction of the whiskers that informs the barrel cortex, the primary mechanosensory area for the whiskers, of which direction the whiskers are moving in so that it can correctly interpret any mechanosensory input onto the whisker. So this could be very important because getting a mechanosensory input onto a whisker when you're pushing it outward versus when something's coming toward your face and deflecting it is a very important distinction for a mouse to make. So finally get to worms and worm navigation. You're probably all familiar with sort of the basic features of worm locomotion and how they navigate in gradients, but I'm gonna review it here to make a couple points relevant for this talk. These are worms sped up navigating toward a point source of isoamyl alcohol, so something that smells good. And they're very good at it. That whole thing was 3 or 4 minutes, except for this guy. And it's thought that most of this is done through executing a biased random walk strategy. And so this is basically consists of forward runs, and then the animal modulates how often it performs these pirouettes. So these are sequences of reversals and turns that serve to pseudo-randomly reorient the animal. So this comes from work by Howard Berg, who described this strategy in bacteria, and Jonathan Pierce in Sean Lockery's lab, who showed that this is the principal mechanism that worms use for salt chemotaxis. And so where we look at where these animals execute these reversals and turns, we can see they're clustered together into these pirouettes. They tend to occur when the animal's further away and not moving up the gradient. And it's hard to see in a static image, but they occur when the animal is moving in the wrong direction. And so what's happening is the animal is integrating over time. And if the environment's smelling better and better, it suppresses the probability of a reorientation. And if the environment's smelling worse and worse, the odor's going away, it increases the probability of a reorientation. So once the animals are all oriented in generally the right direction, you can see they effectively stop reorienting until they reach the peak of the source where they stop because they're dead. But that's not all worms do. And we've seen for a long time navigation behaviors that clearly don't rely on the biased random walk. And the most striking of these, I think, is this isothermal tracking behavior. So these are tracks of animals who are in a radial temperature gradient. So these plates were cold in the center and then warm on the periphery. So there's this sort of these bands of isotherms going out from the center. And you can see that animals choose a preferred temperature, and they do these long forward runs where they track these temperatures very precisely without ever reversing or correcting course, or very rarely doing it. So this requires that they continuously steer. So this is fundamentally different from the biased random walk. And then Yuichi Ino noticed some years ago that often animals will do this during salt chemotaxis as well. And since others have shown that it happens in response to odorants and food and all sorts of things as well, animals can steer. And we can see it in this assay where this animal is off course, and where it reaches a point where it's running almost orthogonal to the gradient, it corrects course and steers gradually up the gradient in the right direction. So navigation is a combination of these two modes of wayfinding, this turning model or clinokinesis, and the steering model or clinotaxis. And these probably predominate under different conditions. This one where the animal can sense over tens of seconds what's changing the environment, and this when animals are running orthogonal to the gradient. You can see the curving is strongest at a 90-degree bearing here. So how might they do it? Worms obviously don't really have stereo senses, although they have pairs of neurons, because all the sensory organs are effectively in a point at the tip of the nose. So there's no obvious way for them to detect a spatial distribution when not moving. However, the worms are usually moving, especially when they're chemotaxing, and they wiggle their nose back and forth. And so this is an opportunity to acquire information about the spatial distribution of a stimulus, and it seems like the most plausible mechanism for determining the direction of a gradient and steering in the right direction when you're running orthogonal to the stimulus like this. So in order to do this, the nervous system needs to have information about head position and information about odor, in the case, all the cases I'm going to show you, but it works for a variety of stimuli. Somewhere it needs to bring these two pieces of information together and convert it into a modulation of the motor system to produce this curved trajectory rather in the right direction. And so that's the question we've been interested in for some time now. Salcato, when he was in Corey Bargman's lab, showed that sensory neurons have the temporal features they need to encode stimuli across head sweep. So this is isoamyl alcohol and calcium and AWC sensory neuron, showing that if you flicker the stimulus at 1 Hertz, which is about the frequency of head movements, AWC is really good at high fidelity encoding of those signals. And then furthermore, in a really cool experiment, he showed that you need that temporal sensitivity in order to steer. So in this graph, we're looking at the black and the blue trace. He used the odor-3 mutant, which doesn't prevent AWC from responding to odor, but it slows it down. So there's a lag of about a second from when AWC would normally respond to an odor to when it actually does. And in those mutants, you lose this ability to steer in a systematic way in response to the gradient. So by introducing this time lag, you throw the system sort of out of sync, and you can no longer detect accurately, we can no longer integrate head position and sensory input accurately. And then Kat McCormick in Sean Lockery's lab used this chip they call the river chip, which holds the worm in its mid-body and allows it to bend its head into one of 2 stimulus streams flowing past it in a microfluidic device. And they showed that animals can, based on an asymmetry in the environment, the sensory asymmetry, they can control their head movements to put their head into a preferred stimulus. So the gray box here is, I think, an attractive concentration of salt. And you can see it preferentially keeps its head to the right side. So they need this information to steer. And in this sort of isolated preparation, they can use that information to orient their head in the right direction. So the pieces are there at the sensory level and the behavioral level. And we're interested in sort of finding out how this information is brought together. So in the lab, animals encounter exafference, so this outside the world. An animal can't control when you're gonna whack it with an eyelash. And in these microfluidic devices we use, its behavior has nothing to do with when the stimulus is going to change. But then there are assays that specifically employ reafference, like the chip I just showed you, where the animal's own behavior determines the chemosensory input it receives, or in these chemotaxis assays using the steering mechanism, where its own movement is what produces the sensory differential across head sweeps. So again, this is how we think it has to work, is that head position and odor are integrated. So to put this in sort of a corollary discharge ethological chart format, here we have imagined central pattern generator for the head that initiates these head oscillations. That's not completely understood. This entrains motor neurons that drive these dorsal and ventral head bends. This leads to the contraction of muscles that actually produces the head bends. And then these head bends are what produce reafference. So in this case, the animal is bending toward a smell and away from a smell, producing this oscillating reafferent sensory input. These same sensory neurons are detecting exafference or anything else that's happening in the world. And this information is being communicated to some interneurons. And then because we believe these 2 kinds of information have to be brought together, these interneurons also have to receive information about head movement, most likely from motor neurons and a corollary discharge circuit, just because that's the most parsimonious way to do it. It's not the only way to do it, but because it's right, it's what I'm showing you. And then these interneurons have to be able to usefully modulate motor neuron activity in order to produce an asymmetric gait and produce steering behavior. So because the worm has this sort of limited neuroanatomy because of the small number of neurons, you can almost look for motifs like this. And because this has quite a few requirements, you have to have sensory neurons that are responsive to all the modalities involved in steering, interneurons that they communicate with, but then also interneurons that receive the right feedback and have the right connectivity to head motor neurons to perform this function. And so in this representation of the worm's wiring diagram, we have sensory neurons here. They're duplicated across these axes, and so these are connections between sensory neurons. These are connections from sensory to interneurons, very dense interneuron connections, and onto motor neurons and so forth. And so you can see there's a limited amount of feedback from motor neurons to interneurons, and it's even more limited if you restrict yourself to head motor neurons. And so this allows us to effectively look for places in the nervous system where this kind of integration can occur. So these are the muscles of the head that control dorsal and ventral head bends. These are the principal classes of motor neuron that contribute to or modulate these bends. And really, there's only one interneuron that sits at the right place to perform that function, and it's an interneuron called RIA, which receives a lot of synaptic input from these SMD head motor neurons, and also has synaptic outputs to the SMDs and another class of head motor neuron, the RMDs. RMDs and SMDs aren't required for head movement, but they're clearly involved in head movement. And at least for the SMDs, their activity is perfectly locked to head movement. So SMDD is active during dorsal muscle contractions, SMDV during ventral muscle contractions. So there's the potential for a corollary discharge signal from the SMDs to RIA to sort of report which way the head is bending, for RIAs to feed back onto these motor neurons to affect behavior. And at the same time, RIA receives input from about 30 neurons that are either sensory neurons or interneurons, first layer interneurons, that are involved in encoding thermosensory, chemosensory, nociceptive information. So these, all the pieces are there. So Exafference and reafferent input comes to RIA as well as all these motor connections. So the question is, how can a neuron, a single neuron, receive these very different kinds of information, encode them in a useful way, and then produce an output that somehow synthesizes those two pieces of information? And for RIA, the sort of key to that question is its subcellular synaptic organization. So this is the original wiring diagram data from White et al. In the 1980s. And you can zoom in and you can go through and count every synapse, whether it's an input or output synapse, and what its synaptic partner is. And if you do that for RIA, you can see there's this very strict segregation of the synapses. So all of the sensory and interneuron, these are just a few, there are about 30, but all those related to sensory modalities synapse onto this loop domain in the ventral nerve cord of the axon, whereas all the connections to the SMD motor neurons are in the nerve ring. And they're further compartmentalized in the sense that the inputs from SMD to RIA are only on the dorsal side of the nerve ring, and the inputs from the SMD that innervates ventral muscles only innervate RIA on the ventral side. So there's this sort of anatomical clustering of inputs related to sensory motor function that could relate to how RIA keeps these kinds of information separate. And if you look at what RIA does as the animal moves, you can see immediately that these compartments do have some functional significance. Again, these are those three compartments and what they look like in a single focal plane of an RIA neuron expressing GCAMP. And you can just see by eye that there's this sort of relationship between the GCAMP signal in either the dorsal or the ventral part and what the head's doing. Of course, just seeing it isn't enough, although it was a pretty exciting day when we saw it. We have to quantify it, and we do that using cross-correlation. So we look at how these two time series align with each other in time, head movement and the calcium signal, and we defined ventral head bends as positive, so there's this positive relationship that's not super strong, but pretty good. between, between calcium in the ventral part of the nerve ring and head bends. And because we defined dorsal head bends as negative, there's a negative correlation between head bending and calcium signal in the dorsal part of the nerve ring. So clearly these compartments are possibly, among other things, because of the sort of moderate nature of the correlation, representing something about the position and or movement of the head. The SMDs that innervate the head muscles are cholinergic motor neurons, and so we started looking for candidate acetylcholine receptors on RIA, and we found that one called GAR3, a muscarinic acetylcholine receptor that's sensitive to scopolamine, is the receptor in RIA that's responsible for producing those signals. So if we look at a GAR3 mutant, we lose all the correlation between head movement and RIA calcium dynamics, even though the head keeps moving, because again, the SMDs aren't essential for head movement, and neither is RIA. And EGL8, which is the phospholipase C-beta homolog downstream of this GPCR, in those mutants, you also lose the correlation completely. Another muscarinic receptor, GAR1, has no effect. And if we express a GAR3 cDNA specifically in RIA, we recover some of this dorsal ventral correlated activity. So we feel pretty confident that GAR3 is a receptor in RIA that mediates this motor encoding. This IP3 receptor is not the right receptor, and we still don't really know the source of the internal calcium store that produces these local calcium events. So that's the motor encoding side. We have these motor neurons that control or contribute to muscle contraction in the head, and they produce what we think is a corollary discharge signal to RIA that produces these local responses in these domains of the axon. So the other side, of course, is the sensory side. All of these synapse onto the loop, so we start looking in that domain for interesting sensory responses. And these are heat maps that show movement of the head, the presentation of the odor, the removal of the odor, and then the calcium responses in that loop domain. And we can see there's this pretty good relationship. When the odor is presented, there's this sort of global inhibition of calcium levels in the loop compartment. They're higher during odor presentation for reasons we don't understand. And then upon odor removal, there's this sort of calcium jump. Not all the time, but in a lot of cases. What's interesting is that this pattern of sensory response is not just in the loop domain, it's also in these nerve ring domains that I just showed you correlate well with head movement. So this might be partly why the correlations are only partial, is that in addition to responding to head position and movement, these nerve ring compartments are also clearly responding the same way the loop does to sensory presentation and removal. And so we think that there's this sensory encoding where the presentation of the odor, the attractant odor, leads to a whole axon inhibition of calcium. The removal of the odor produces a whole axon influx of calcium in response. And so these two types of calcium signal are additively combined. And if anyone's interested, I can tell you why we think it's additive as opposed to mutually exclusive or antagonistic to encode these two kinds of information in one neuron. So to look at how they turn this into behavioral responses that underlie head orientation behaviors, we went back to looking at behavior in wild type versus the GAR3 mutant, where we'd impaired the motor encoding aspect of RIA. And overall, GAR3 animals look pretty well. But if you look at them more closely, they have this kind of, this is a wild type animal on the right, GAR3, look a little slow, and they have this kind of exaggerated gait. So their head bends are much bigger than normal, which you can see if you kind of do this superposition of their body. Wild type have this nice elongated kind of shape, and these are kind of exaggerated large head bend leading to a large gait amplitude. And so we think that what that means is that these motor encoding events are part of a sort of negative feedback system that limits the extent of head bending. Because when you lose it, you have these much larger head bends in both directions. So When you engage this local compartment, you produce, with some delay, a negative feedback signal to the ventral motor system to limit bends in that direction. And then the same thing happens on the dorsal side. So in terms of steering behaviors as a whole, here we're going to look at the tracks of animals. So this is what an animal would look like, and this is sort of a path of its centroid. And wild-type animals look like this in an isothermal tracking task. They sort of find the temperature they like and then do these sort of long curved trajectories and produce tracks that look kind of like this. If we look in a GAR3 animal, we can immediately see there's something wrong, maybe a lot of things wrong. They don't produce these long isothermal tracks. They produce these tightly coiled kind of curly Q tracks. And we don't know quite how to interpret that because they don't do it in a when they're not in a gradient, but clearly something about being in a gradient is making them attempt to do some different behavior, but not be able to track the isotherm. So again, it's the loss of information about head position makes them unable to track a stimulus. And then we put them in the river chip that had come from the Lockery lab and saw the same result that wild-type animals will restrict their head bends to sort of the good-smelling side of the chip, but an animal where a synaptic release from RAA has been blocked using tetanus toxin, so that RAA can no longer influence the head motor system, no longer makes any distinction between these two. And we know these animals can still sense the odor, and in fact, they can do chemotaxis to an odor using a biased random walk, but they can't orient their head, at least in this device, in response to an odor. And this is a quantification of that, again, with the RAA tetanus toxin. And then this is scopolamine, the drug that blocks the muscarinic receptor on RAA. So you need both input and output from RAA to orient your head relative to an asymmetric stimulus like this. Okay, so how does steering work in our little cartoon model of this? What we think is happening is these two sources of calcium are interacting in such a way that you basically break the symmetry of this negative feedback system. So when you bend in a good smelling direction, you're engaging the negative feedback, but because the good smell globally suppresses the neuron, it's weakened when it goes in that direction. And when you bend away from the good smell, you're globally exciting the neuron, so you get a stronger negative feedback signal in the less preferred direction. So you limit head bends dorsally in this case, you extend head bends ventrally, and you produce a asymmetric gait and therefore a curved path in the ventral direction. So limited evidence for that, but that's our working model. So putting it together in another way, looking more at like a sort of signal processing in the neuron way, we know that as the animal crawls forward, it's producing these anti-phase signal in NRV and NRD because each compartment is correlated with a different head direction. And when you add a stimulus on top of that, because of the movement of the head, that stimulus is going to be in phase with one of the compartments and always be out of phase with the other compartment. And so you'll have, in this case, an additive effect with the dorsal compartment and a canceling effect with the ventral compartment, and again, leading to an asymmetric output from RAA, an asymmetric gait, and a curved trajectory. And so this is how the reafference gets combined with head position information to produce a steering behavior, we think. So there's a question here still about how the animal distinguishes reafference from exafference, and that's because if RAA is responsive to odors no matter where they're encountered, the animal can't really be sure that a stimulus is because it's head bent, or if it's just a stimulus that's appearing because it ran into something or something an experimenter happened to dump on it. And so we started looking at how sensory responses relate to head position at the time of odor presentation or removal. And so we're looking at the same heat map, but here we're sorting all of these trials by where the head happened to be when the odor was removed. So again, it's arbitrary in the experiments, but we can sort them. So these are cases where the head was bent ventrally when the odor was removed, where it was effectively not bent, and where it was bent ventrally. And if we sort the responses the same way, we can see there's this very clear relationship where a bent head produces much more reliable and perhaps larger calcium response is in the loop, whereas a response is much less likely or smaller when the head is not bent. So there's this relationship between the posture of the animal, or the position and movement of the head, and the likelihood that RAA is going to be responsive to a change in an odor. And we can look at this in a different way, just looking at position of the head at the time of odor removal, and then the average size of the response. We see this very clear response. So how is this happening? It occurred to us that maybe the motor encoding signal we see is actually mediating this. So we have this motor encoding signal that tells the neuron when the head's bent, and perhaps these local signals are somehow modulating the magnitude of this whole cell sensory response. But that turned out not to be true. When we look at the responses, this is head bent and unbent in a wild type. All responses are larger in GAR3 mutants for reasons we don't know, but this relationship still exists, that the responses when the head is bent are still much larger than responses when the head is not bent. And so it can't be that these motor encoding signals from the motor neurons are what is sort of filtering sensory input to RAA, so there has to be another mechanism. However, we wanted to see if there were head orientation behaviors specifically related to this question of posture at the time of odor removal. So we looked at animals under 6 conditions: the presentation of the odor, no change in odor, and the removal of the odor, either when the head was bent or when the head was not bent, and look at the response, the behavioral response in terms of head velocity of the animal when that occurred. And so in this case, positive velocity is deflection in either direction, and negative velocity is movement toward the center. And it's really only if the head is bent and you remove the odor that you produce a reliable behavioral result, and that result is a rapid retraction of the head from the direction it was bent in. So that rapid withdrawal of the head, so this is animals without their head bent, and they show no withdrawal of the head. So these are small positive values in the change in velocity. Wild type animals show these rapid withdrawals, so these large negative velocities, only when the head is bent and when the odor is removed. And you lose this behavioral response in a GAR3 mutant. You get it back if you express GAR3, specifically in RAA, and you lose it if you block synaptic output from RAA. So although this GAR3 motor encoding isn't required to gate the sensory information, it is required to produce a behavioral response to a change in sensory information. So RAA seems to be sort of a central switch point for producing that behavioral response. So that led us to what we call a gate and switch model for RAA function, where when the head is not bent, the gate is effectively closed, and RAA ignores changes in sensory stimuli. This requires that we invent another element here, sort of putative proprioceptor, that can be another source of information about head position to control this gate, right? And Lily Guinet has found one mutant that seems to eliminate the difference in sensory encoding. So these are sensory responses in RAA when the head's bent, when the head's not bent. That's giving us a clue about what neurons might be involved in this second head movement encoding pathway. But in either case, under normal conditions, when the head is bent, this gate is open, RAA is responsive to sensory input, and then these motor encoding compartments sort of route this signal either in the dorsal or in the ventral direction to limit head bends in the non-preferred direction, and leads to an asymmetric gate and output. So what we think is that like in these other systems where these sort of active sensing behaviors modulate sensory perception in a useful way, we have this motor input to RAA that comes from oscillatory bending in the head, and this modulates responses to chemical and thermal stimuli in a way that can produce a gate asymmetry that steers the animal in the right direction. Okay, this is the final sort of ethological model of that, and the final points are that they are able to integrate head position and sensory input to detect the spatial distribution of the stimuli. A single interneuron encodes these through separate physiological pathways. And the big picture, I think, to me, and what's interesting is that these kinds of self-representations that occur in RAA, at least in terms of the motor encoding, are fundamental to accurate sensory perception, even in an extremely simple system like this, which was perhaps underappreciated because these are normally talked about in the realm of very sort of sophisticated sensory motor and cognitive behaviors in larger animals. Okay, this is the lab at the moment, and this work that I showed, some of it was begun in Yoon's lab when I was a postdoc, and more recently all this work has been done by Marie-Helene and by Lily, and these are the other members of the lab and our sources of support. And thanks very much for your attention. Happy to take questions.
Okay, thank you, Michael. So we have a first question from Daniel Wittviet. Since RIA ventral domain makes synapse to both dorsal and ventral motor neuron, and RIA dorsal domain also makes synapse to both, how do you think RIA control if ventral or dorsal motor neurons are regulated?
Yeah, it's a great question. It bothers me a lot. But I think if you look at the anatomy of the microcircuit, it's true that the ventral domain has outputs to both SMDD and SMDV, but at very different sites on those neurons. So the SMDs have clusters of synapses near their cell bodies, and they have clusters of synapses near the neuromuscular junctions. And my best guess right now is that those inputs from RAA are functionally different. So we're looking at different classes of receptors on the SMDs. And so I think that it's possible that one cluster of those receptors is excitatory and the other is inhibitory. And so that it's providing one output, which we think is glutamate from RAA, that's having opposing effects on the dorsal and ventral motor neurons based on which set of synapses on those neurons it's activating. I think I probably spiraled into nonsense there, but I have pictures I could show you that make sense. So you can email me if you want to.
All right. Next question from Michael O'Donnell. Great talk. Presumably, preferred direction can be either dorsal or ventral depending on the animal orientation. Do you think the state of the motor neuron postsynaptic to RIA play a role in discerning the ventral dorsal bias?
Yeah, that's another, that's an idea that Sean Lockery and Eduardo Izquierdo published a while ago, which is that the motor neurons or even the muscles themselves could be differentially responsible to a signal depending on their own activation state. And that alone, in theory, would be enough to give you an asymmetric response to a symmetric output from a neuron like RAA. We don't have any evidence of that because we don't know what any of the receptors are on the motor neurons yet for the signal from RAA. So we're trying to look at those, and we're trying to directly visualize synaptic output from RAA, and that should address those questions somewhat, but that's right, we don't know.
Okay, next question from Philip Kidd. I'm curious what's known about the biophysical mechanisms that allow RAA to maintain semi-separate compartments.
Yeah, so we think that the local signals in the nerve ring are not due to membrane depolarization because for reasons that are barely comprehensible to me, but that Sean and Miriam Goodman explained to me, it would be very hard in these neurons to have a section of the axon that was electrically different from a different part of the axon. So we think that there's some kind of local calcium store in the case of the compartments, but in the case of the loop, these sensory responses are very easy to interpret as a global opening of voltage-gated calcium channels. In terms of maintaining the spatial restriction of the compartments, we do have some ideas. So Marie-Helene is working on some mutants that lose this compartmentalization, and we've seen, and she's identified some calcium regulatory proteins, including some active calcium extrusion pumps that seem to restrict these compartments and maybe the temporal features of these events as well. What's really cool about those is some of them are things that sculpt sort of calcium responses in dendritic spines and vertebrate neurons. So it suggests there's this deeply shared set of mechanisms for regulating calcium compartments in all animals.
Okay, then I think it's next question from Troy Diarmid. Similarly, if you force optimal RIA activity time, for example, manually or with optogenetics, can you promote advantage reorientation regardless of the head position?
Yeah, that's a good question. And to do that, we really need, like you say, some kind of access where we can control RIA responses or the activity upstream of RIA in a closed loop kind of way. So we're working on that, where we have optogenetics set up where there's real-time feedback of the animal's head movements and posture that control the optogenetic stimulation. So that's the question, exact question we're trying to answer with that setup.
All right, and then last question for follow-up from Daniel Viviet. Why do you think there are both SMDs and RMDs? Because their function seems to be redundant.
Yeah, so there are lots of head motor neurons that seem to have overlapping functions and partially redundant functions. The RMDs are frustrating in that I've never seen a calcium response in an RMD with quite a bit of looking. So RMDD and RMDV, you can see some activity in RMD itself. There are 6 of those. So I don't really know what RMD is doing. The other frustrating thing about even the SMDs is that we're fairly sure that along with the RMDs, they're the effectors of RIA's output. But even when we drive RIA activity with a fast sensory stimulus, so we're really driving RIA off and on every couple of seconds, and we see these really robust calcium responses in RIA, which we would like to believe are driving synaptic output from RIA, we don't see anything happening in SMDs that correlates with the activity in RIA. SMDs are just completely locked to head movement. And so my best guess there is that when we look at SMDs and RMDs, we're only looking at somatic calcium, so in the cell body, because we don't have, or at the time, we didn't have specific promoters for them. And I think that maybe what's happening is the SMD bulk calcium is just phase locked perfectly to head movement, and that the input from RIA is doing something modulatory more locally out at the neuromuscular junction. So perhaps glutamate's being released onto either a metabotropic glutamate receptor or a glutamate-gated chloride channel, and that's affecting neuromuscular junctions of SMDs. And we have some evidence for that because we have other behavioral assays for RIA dysfunction, and some of the GLICs and mGlu-R mutants have defects that look the same as RIA defects. So we think it's a promising route.
All right. I think that's it for the question. Thank you both to you and Javier for joining us today, and to everyone who's still in the listening. And I will see you all next week for our next session. Thank you very much for joining.
Thanks, everyone.
Have a great week.